Here, do you see my point?
Yes,
Yeah.
perfect. Yeah.
So whenever you're ready, so just let me know. So so Dimitri, do you want to get it started now?
As you wish.
Okay, cool. So let's get started then. So hi everybody. So once again, it's a pleasure to welcome you all to attend this the webinar series. So today, it is a great pleasure to have Dr. Dimitri van der Wiel to give us a talk on GSP for neuroimaging. So let me just first try to introduce the speaker a little bit. I'm pretty sure that a lot of you know him already. But just let me read off from his bio. So he received his PhD degree from computer science engineering from the Ghent University in Belgium, and then after that he was a postdoc fellow at the lab of Professor Michael Unser at the EPFL, and then in 2009 he received the Swiss National Science Foundation professorship since 2015, and then he became the associate professor of bioengineering at the EPFL, and also co-affiliated with the University of Geneva. His main research interest is in developing new approaches for computational neuroimaging to advance cognitive and clinical neurosciences using tools from wavelet sparsity, deconvolution, and GSPs. And he received, of course, many accolades, and just let me name a few. He was an elected IEEE Fellow in 2020, and also an SPS Distinguished Lecturer in 2021 to 2022. And he also provided numerous services to IEEE and various journals, such as being a senior editor board member for the IEEE SPM, also an editor for Science Journal of Imaging Science, for TSPs, and also for transitions on image processing. He was also the co-chair of the BISPTS of the SPS Society, and also the founding chair of the EuroSIP Biomedical Imaging and Signal Analytics SAT. So I think we are all excited to hearing Dimitri's talks. So the floor is yours.
Thanks a lot. Thanks for you and Geert for the very kind invitation, and it's a pleasure to be here with you today. So my talk will be on GSP, and specifically how we can use GSP to process neuroimaging data, in particular brain anatomy and activity, and GSP will be the link between them, as you will see during the talk. So let me maybe start with just a very quick historical perspective, because humanity has been for a very long time already looking to map the brain, which refers to the fact that we want to assign specific functions to specific anatomical brain regions. And this idea that there is specialization in the brain dates back to at least 1 millenary, where we had Ibn Sina in Persia, Avicenna, as he was known in the West, who already in his Canon of Medicine proposed this idea that you had specialized brain regions, as you can see here on this picture. And this idea, which is of course a very useful concept, stayed on over the centuries. And like 200 years ago, there was this pseudoscience known as phrenology, where people like Franz Joseph Gall designed a psychograph that would measure the bumps on your skull. And when you had a bigger bump, that would be a sign of having a bigger underlying brain region, and that would have been related to certain personality traits. Of course, we know that this doesn't make a lot of sense today, but the whole idea that you had localized function in the brain was around, and of course stays around into today, because today with modern neuroimaging techniques, we are actually still continuing this quest. And we do have already a good idea of which regions in the brain specialize for which tasks. But we also have understood that the brain is more than just the sum of its parts. And there is a whole integrated organizational principle, let's say a networked system that is also very clear from today's research. So the imaging modality of choice for this talk will be magnetic resonance imaging, or MRI. And MRI has been made available thanks to major scientific and technological breakthroughs, as kind of shown by the fact that not less than 8 Nobel Prizes can be linked to MRI, which is really a lot. But it's, of course, a truly magnificent imaging modality. It allows non-invasive imaging of the human body and the brain in particular, not only in terms of anatomy, but also in terms of function or activity, if you want. And one of the beautiful properties of MRI is that it's using a so-called endogenous contrast. So you don't need to inject anything. There is no radioactive tracer, like in PET, for instance, but you are actually just using the natural distribution of hydrogen throughout the body, which is abundant, of course, because there is a lot of water in our tissue that we have. And I guess everybody knows MRI for its beautiful structural images. Here you see a whole brain structural image, which is, of course, extremely important in clinical practice. You can go down to submillimetric spatial resolution today, but actually there's a lot more in MRI than this type of images. And I will show you in the next slides what type of MRI data we are using to do graph signal processing. So I will show you what type of MRI neuroimaging data we will use specifically to build graphs, brain graphs, and then to consider graph signals on top of those brain graphs. First of all, the brain graphs. So next to those anatomical images that I showed you before, you can also use MRI to acquire so-called diffusion-weighted volumes. And those volumes, and let me maybe use the pointer, they actually allow you to make the signal sensitive to certain orientations in the tissue. And this is useful because in the gray matter, where most of the cell bodies of the neurons are located, so for instance, could be in this red spot over here, you have outgoing axonal bundles that reach out into other gray matter regions. And those axonal bundles, they need to run through the white matter, where they actually organize and group together, as you can see here from this microscopic zoomed image. And so at the millimeter scale, these orientations are still strong enough such that they actually alter the signal of this diffusion-weighted MRI. And what this then allows is to, out of this type of data, reconstruct like a virtual streamline or virtual reconstruction of the axonal bundles, which you can think about a little bit like a virtual dissection of the white matter. And out of this tractography type of data, one can then superpose, by the choice of an anatomical brain atlas, the knowledge of the position and the shape of different brain regions, which you can see here illustrated for one type of atlas. So given the number of streamlines that run between those brain regions out of the tractography, you can then characterize the structural connectivity strength And that can be all combined into a graph representation where the nodes will be the brain regions and the edge weights that you store in a weighted adjacency matrix will be extracted out of these streamlines. And if you put it all together, this adjacency matrix might look like the one over here, which we refer to as a structural connectome, or if you like, a wiring diagram of the brain. And in this case, the atlas was the so-called Glaser atlas, which contains about 360 regions, so 180 regions per hemisphere. So that's the brain graph. Now, there's yet another type of MRI imaging data we can use, which we know as functional MRI. And functional MRI, as the name says, is aimed to recover activity, brain activity. Now, let me explain a little bit how this works. So you see that now we have a series of fMRI volumes, and we can still do whole brain coverage, and we can still get close to millimetric spatial resolution. But of course, compared to the anatomical images I showed you before, you can see that the quality, the spatial quality here is much less. Now, the point is that here you want to go fast. So we will do one volume about every 1 to 3 seconds. And we actually are interested in how the signal at every spatial location is changing over time. And the reason that we are interested in that is because these fMRI values, they reflect the blood oxygenation level dependent signal, or shortly known as BOLT. And the reason that is interesting is because there is the so-called neurovascular coupling, which tells us that given sufficient initial neuronal activity through neurovascular coupling and the vascular response that follows, we will have a change in the fMRI signal known as the BOLT signal over here. So basically, BOLT fMRI is a slow proxy for underlying neural activity. And so that's the reason that this type of MRI images are called functional. Let me show you here in little visualization how this data typically looks like. So what you see here at the top left is a visualization with some minimal pre-processing of so-called resting state fMRI. And the coloring is made such that whatever is more red is higher with respect to baseline, and blue means below baseline. And the video is a little bit accelerated, so in reality, these changes of the BOLT signal are slower, but you can see that you have a lot of fluctuations and that they are mostly very structured. You see that they're usually symmetric between the hemispheres, and so a lot is going on, although this subject is in principle doing nothing, so but which actually means a lot in the brain. Now, one way we can process this type of fMRI data is again bring in the knowledge of our anatomical atlas and then extract for each of the brain regions the regional average time course. And so those time courses can be obtained, and then you can, well, what we will consider next will be the graph signal, which will be nothing else than for each time point here, the spatial pattern of the fMRI BOLT signal. So in other words, the full data set will now be a matrix X, where we have the number of rows being the number of regions and the number of columns being the number of time points, which can also contain subjects if you concatenate them, for instance. So a graph signal will be a column out of this matrix, which is a vector of length n. And once we have the brain graph and the brain graph signals, we have enough, let's say, to start playing with graph signal processing with GSP, which has been a very prolific area in the signal processing community. And I just flashed here a couple of very nice covers from IEEE Signal Processing Magazine, also proceedings of the IEEE, where many groups have been thinking and proposing about re-engineering somehow classical signal processing operations in the context of graphs. And some examples are Fourier and wavelet transforms, but of course also sampling problems and signal recovery, and so on. So this is a very rich new subfield in signal processing. And if you're interested, I certainly refer you to these special issues, which are very interesting. Now, for the purpose of this talk, I will focus on a few of these topics and for what we have used them already to process neuroimaging data. So the first thing I like to introduce is the graph Fourier transform, which basically brings together our dear friend Euler, by the way, who's Swiss, and who's seen as the father of graph theory, and see how he can basically meet with Fourier, the father of harmonic analysis. And to let them meet, we first need to look at the graph Laplacian, defined as the degree matrix minus the adjacency matrix. And then we're going to introduce the eigendecomposition of the graph Laplacian, which is going to actually define our graph Fourier transform. Because of this eigendecomposition of the graph Laplacian, we will consider the eigenvalues lambda as the as encoding for the graph frequency, and then the eigenvectors as the graph Fourier vectors. So if this is the first time you see this, let me perhaps show you an analog with how this looks like if we apply this recipe to a cycle graph. And you will see that in that case, the graph Fourier transform becomes, in essence, equivalent to the discrete Fourier transform. So the cycle graph is shown over here. It's basically a model for a one-dimensional signal with periodic boundary conditions, and its adjacency matrix looks, of course, like shown over here. So we can build the graph Laplacian for this simple graph, and then we can compute the eigendecomposition. And what you can see is that indeed we have eigenvalues that increase, so increasing frequency. And if you look at the associated eigenvectors, you see that we recover a very similar structure as the DFT, where we have a first eigenvector with a zero eigenvalue, that is the constant, and then we have actually two eigenvectors encoding for a low frequency with the same eigenvalue. So you can think about these cycle cosine and sine, and then you go to higher frequencies and higher, and the last one is actually the Nyquist frequency, as you see in the last column of this matrix. So that's kind of reassuring, and I can already tell you that this recipe will work for brain graphs. But before doing that, let me also put a little warning here, because this very nice interpretation of the eigendecomposition of the Laplacian as a suitable graph Fourier transform is not always actually valid. And if you think about a few more pathological graphs, by which I mean graphs that are much more irregular, then this nice interpretation doesn't actually work anymore. So Euler and Fourier don't meet. For instance, if you have a complete graph, which is, you can think about overconnected, then we know that all the eigenvalues will be 1 except 1, 0, will be n except 1, 0. If you have a star graph, you have one eigenvalue 0, 1 n, and all the others 1, so which also doesn't show a very nice harmonic representation. And you have other special graphs like star-like trees and things like that. So you kind of need for this GFT interpretation a sufficiently regular graph, which luckily for us, a brain graph will do. Now, before getting back to the brain graph, let me now introduce how we actually want to use the graph Fourier transform, because in the end, we want to define suitable graph filters. And the ones that we will consider will be a class of filters that are shift invariant under the graph Laplacian. So in other words, such a graph operator H will commute with the graph Laplacian L. And what happens then is that any of those graph operators can be written as the matrix polynomial of the Laplacian. And so, of course, this could be one way of trying to design a filter, which you can then apply to a graph signal to get it filtered. However, if you now bring in the perspective of the graph Fourier transform as the eigendecomposition of the Laplacian, so then we can use this to compute a spectral representation of a graph signal. And then we can also look at what will be the spectral representation of the graph operator. And now, because of this property of our operators H, we can actually quite easily see that the spectral representation of the graph operator will be a diagonal matrix because it's by construction diagonalized by the Laplacian. And so that means that implementing the filtering in the spectral domain will revert to a coefficient-wise multiplication of our spectral representation of the graph signal. So we simply have a diagonal matrix H hat, which we multiply with our Fourier vector X hat. And once we have done that, we can, of course, get back to the regular domain to have also our same filtered graph signal, but this time obtained through the spectral representation. And this, of course, is very reminiscent of the Fourier convolution theorem, but this time in a graph setting. And actually, by this very simple relationship, as we go through the spectral domain, we can also read out of this combination of GFT with H hat and inverse GFT, the so-called shift variant impulse responses of our graph filters, which are in principle, well, not trivial to see otherwise. And so the big advantage of the spectral idea is that we can now design and implement even our graph filters in the spectral domain by actually directly specifying the spectral specification of the graph filter. Okay, and this can also be a design principle, even if you want to implement it finally in the graph domain as well. And there is a lot of literature, some of it referred here at the bottom of the slide, that proposes very elegant ways to do this. Okay, so just to give you a couple of examples, if you imagine that this is the Laplacian eigenvalues, then you can think about maybe you can specify some low-pass filter like a simple spectral window or a high-pass, of course. You could implement the Laplacian in the spectral domain by multiplying with the eigenvalues themselves, or you could make combinations, or you could do a diffusion filter, which is another very nice spectral window, etc., etc. Okay, so let's now get back to the specific application here, which is for our brain graph. And what you see here are the Laplacian eigenmodes for the structural connectome that I showed you before, as we derived it from diffusion-weighted MRI. And as you can see, we do find very nice eigenmodes in terms of spatial distribution. So first of all, they basically encode for the geometrical organization in the brain. So we get, after the constant, we get a left-right hemisphere eigenmode. Then we get anterior-posterior pattern. And then we start to get more complex combinations that take into account, let's say, more intricate structure of the structural connector. And so we get actually very nicely distributed oscillatory patterns up to the very final eigen modes, where we have a few isolated ones, but really almost all of them are really very nicely defined. So we could say that we have a very good graph Fourier transform definition. So once we have that, we can now use our graph Fourier transform to have a first look at our graph signals. And the very first thing I like to show you is how the graph energy spectral density looks like. So very simple, we're going to take our fMRI data matrix. Every column here, so every graph signal, will be transformed in the graph spectral domain we will compute the energy and we compute the average, and this is basically in a log-log scale what you get. So because the first eigenmodes are much more smooth, as you have seen, they should be because the eigenvalue is also reflecting the smoothness. And so you see that the energy of those first eigenmodes is clearly much higher than for higher frequency eigenmodes. So in some way, brain activity patterns, they naturally respect the smoothness of the underlying structural connector. And that actually gave the idea to now define graph spectral windows, where we would say, let's look at the simple ideal low pass, where we have the low pass eigenmodes that represent aligned, that when we filter with that would represent activity that is aligned with respect to the underlying brain structure. And the complementary high-pass filter would, when we apply it to fMRI data, would reveal the activity that is more liberal with respect to underlying brain structure. And the cutoff frequency between those two filters would be defined by having basically a median energy split. So in principle, we expect in total as much energy in the low pass than in the high pass on average. And so here you see a representation for a segment of the data where you can see aligned, much more smoother indeed, and then liberal signal, which is much more salient as we expect. So given this interpretation that low pass relates to aligned signal and high pass to liberal signal, we can now, for each and every region out of these two filtered signal representations, compute what we call the structural decoupling index, which is the ratio of energy in the liberal versus the aligned signal. And you can do that, of course, for every region in your that you consider in your connector. And so this is what we call the structural decoupling index. If you do that and you represent that into a brain view, and here the color coding is such that blue means more coupled, so the structural decoupling index would be smaller than 1, and red would be more decoupled, so meaning that, of course, the SDI is larger than 1. And so what we get is a map that has some striking spatial structure. In particular, we have brain regions that are much more coupled in the visual cortex, in the auditory cortex, and the somatomotor cortex. And they really stand out with respect to the others, which are either, let's say, not without any preference. Those are more greenish. And then we have other regions which are more reddish, orange-reddish, which are actually strikingly more decoupled in our data than we actually would expect if structural decoupling would be uniform across the brain. Now, of course, when you see that map, from a neuroscience perspective, it's very striking that those regions that are more coupled are lying in the sensory cortices. But you know how much coupled is actually, significant. And that brings us to an opportunity again for graph signal processing to bring in null models for statistical testing. And the aim here is to check what is the influence of the true empirical anatomy and activity, and to what extent is it significant in those measures of structural decoupling index that I showed you before. So the first type of null model I will introduce is to randomize the graph signals. So those that reflect activity or function, if you like. And the idea is to generate surrogate graph signals that will have, by construction, the very same smoothness as the empirical signals. And what I mean with smoothness is smoothness as we measure it by the quadratic form of the Laplacian, which is defined, as you know, in this way, or we can start off with the quadratic form itself. And in the spectral domain, this is actually summing over the energy of the spectral coefficients weighted by the eigenvalues of the graph Laplacian. So, of course, for eigenvectors, we get exactly the eigenvalue, which is always nice to see as well. So this is our measure of smoothness. Now, how could we generate surrogate graph signals that have exactly the same smoothness as the empirical signal? Well, a very simple trick is to take our empirical graph signals, transform them in the graph spectral domain, and then randomize the sign of the spectral coefficients by multiplying randomly with +1 or -1. And so that gives us a surrogate spectral representation, which we can then reconstruct back in the original graph domain. And by construction, it's trivial to see that this simple recipe will preserve smoothness, because if you look at the quadratic form of a surrogate graph signal, it is by construction the same as the smoothness, the quadratic form of the original one, because we are only looking at the squares of the spectral coefficients. So the sign simply doesn't fit. And so the way this works is if you look again at the data matrix, so we bring every column into the spectral domain, we randomize the signs, and then we go back to the original domain, and we have a surrogate data set at the output. And by construction, the energy spectral density will also be the same, for the same reason as the smoothness is preserved. And then, of course, you can repeat this procedure many times to build a whole set of surrogate data sets from which you can derive whatever metric you want to do to build your node distribution. So this is really the recipe that underlies a non-parametric statistical testing framework. And another very nice feature I like to mention is that because we keep this P matrix the same for all time points that we process to generate one surrogate data set, then actually it's also very nice to see that basically the second order statistical structure is completely preserved. So what I mean, if you compute for a surrogate data set x tilde transpose x tilde, that is actually exactly identical to x transpose x, again, because those signs actually cancel out. So it means that, in essence, correlation structure between time points is completely preserved in the surrogate data set that you generate. And this very nice scheme dates back to Elvira Pirondini, who was a postdoc in the lab a couple of years ago, and who's now a faculty at the University of Pittsburgh in the US. So this is the way we can generate surrogate graph signals. There is something more we like to assess, which is the influence of the anatomy or the structure, if you want. And for that, we need to kind of knock out the effect of the structural connectome. So what we're going to propose there is the so-called configuration model, which is out of the empirical structural connectome. We look at the degree distribution. So you can look, for instance, you can look at the degree vector, which is the diagonal of the degree matrix. And then we basically do an outer product of the degree vector properly normalized to come up with the configuration model, which has exactly the same degree distribution as the empirical structural connectome, but which has no preferential connectivity, as you can see, whatsoever. So this is or null for structure, if you will. Okay, so now that we have those new tools to build null models and do statistical testing, we can go back and build again some SDI maps, but this time based on surrogate data. And so if I show you the SDI map, where we have randomized both structure and function, and then we compute an average SDI map, this is what you get. So everything is extremely decoupled, which makes sense because now we have surrogate data that has been generated with the configuration model. So basically, the surrogate data doesn't see the real connectivity in the structural connectome, and therefore, when you measure it with SDI, everything looks very decoupled. There is basically no respect in the surrogate graph signals of the underlying structure. So this is a very nice sanity check, but not so surprising. If you actually compute the structural decoupling index for surrogate data that has been generated with the true empirical structural connectome, you get a map that looks already much more like the one I showed you before. So there is clearly this pattern of more coupled into sensory regions to more decoupled into high-level regions, which is already present. But if I show you again the structural decoupling index for the true empirical data, which has both the original empirical structure and activity in it, you see that this pattern is further reinforced. And so this is interesting because it means that this spatial gradient is somehow already present in surrogate data that is sufficiently realistic in the sense that it has the knowledge of the underlying structural connectome. And preserving the smoothness, but otherwise being random activity patterns, is sufficient to already bring out this general pattern. So I think that's interesting because it means that there is a very strong influence from the structural connectome on this property, although it is further reinforced if we look at the true empirical data. And then, of course, we can do statistical testing. And here you just see the difference between the empirical SDI map and the surrogate SDI map. And you see that indeed there is more coupling, and this is statistically valid in the auditory and the somatomotor regions. There is certainly more decoupling in the parietal and the frontal regions, and also temporal, especially inferior temporal. And then there seems to be a bit more decoupling also in the visual system, so which is maybe not concordant with the other sensory regions, but which is actually confirmed by other work that talks about secondary gradient in terms of hierarchy in brain regions based on other pieces of data, such as functional connectivity and genetic analysis. So maybe as a final word on this is, when I showed you this map, I referred already to the fact that we have kind of a spatial gradient from low-level sensory to high-level transmodal or more association regions. And this can actually be checked to what extent this also matches with knowledge from task fMRI, which has been collected and put together in large databases. And so if you basically rank the regions from being more coupled to being more decoupled, and then you look up those regions into what is the most popular topic in terms of task fMRI findings, you get an association matrix like the one at the right-hand side here, where you can see that regions that are more coupled do indeed refer typically more to or pop up more in studies related to visual perception, motor, eye movements, so rather relatively low-level cognitive functions. And if you look at the more decoupled brain regions, they are associated with topics that are much more high level, such as social cognition, emotion, decision-making, and so more advanced memory, etc. Okay, so what I showed you up to now was basically this organization of the structural decoupling index as we can extract it from a group of subjects, which was basically about 100 subjects taken from the Human Connectome project. But the question we had is, okay, right, we do see this very strong spatial gradient in the brain in terms of how activity is, how activity is connected with the underlying structure and how that is quantified by our SDI. But if you would compute it at the level of an individual subject and maybe of an individual run, is there also meaningful information in there? And so what we did to that purpose is we looked in more detail at the subset of the Human Connectome project data, where we have 100 unrelated subjects. And for each of them, we did not only look at the resting state, but we also looked at 7 more task fMRI data runs, basically. And then we can compute for each subject, each task, we can compute the corresponding structural decoupling index map. And then we can ask 2 questions. We could ask the question, to what extent does the structural decoupling index map reveal information about the identity of the subject? So which is a brain fingerprinting task. And the other question we could ask is, to what extent do these structural decoupling index maps reveal information about the type of task that the subject is doing, which is basically task decoding. And so we computed all those SDI maps, and then we started with the first statistical analysis, where we looked at the influence of the subject versus the within subject task factor. So if I first show you here at the left-hand side, the effect of fingerprinting, the regions that are more colored red here correspond to the regions that have a stronger fingerprinting effect. And you can see that those regions are actually mostly the regions that are that had a stronger decoupling before. So which somehow is very satisfactory in a way, because it will tell us that regions that are more high level are actually those regions that are most unique to each and every person. And this is nicely complemented by the analysis. Oh yeah, and to conclude first on fingerprinting, so this is just a statistical map, but if you do a real decoding with a very simple decoder like an LDA, you get already an accuracy of 98%, where chance level is at 1%, in identifying an individual correctly, only based on his SDI map. So this is quite spectacular in terms of specific information that is also encoded into this brain map. And then the second question we asked is to look at the effect of task decoding and how this is encoded into the SDI map. And what you see now is that the most significant regions are related to the sensory cortices. So again, visual, somatomotor, and auditory clearly very strongly pop up. And if we combine this in a real classification approach, we get to an accuracy of about 75%, where chance level is at 12.5 because we had 8 tasks to decode. So there is certainly more in these SDI maps than just a general, organizational trend of that the spatial gradient showed us in the beginning. So to conclude, let me maybe say a few words about what are our ongoing and also our future views on using GSP for neuroimaging and for brain analysis in general. So one thing we are actively working on now is to basically get rid of the anatomical atlas that we chose in the beginning. Those atlases, they typically have about 100 to maximum 1,000 regions. Of course, they are very useful, and they bring they make it easier to localize things in the brain and to reduce somehow your spatial dimensionality in a certain way. But instead, we want to really revisit the brain graph and consider now as nodes of the brain graph each and every voxel, actually both in the gray matter and the white matter. And that brings the number of nodes to consider from about 1,000 to close to 1 million. And this is, of course, a significant challenge. So the first thing is, how are we going to build that brain graph, and why is it worth it? Well, we're going to build the brain graph by just actually, from the diffusion-weighted MRI data, do the local, let's say, to derive the local connectivity in the brain graph by just using the orientation pattern that we get out of the diffusion-weighted MRI data. So there will be no need for tractography. Somehow all this information will be contained into the local connectivity patterns throughout the brain graph. And one major advantage here is that, of course, we don't need tractography anymore. But secondly, there is actually now a meaning of the number of hubs when you travel the graph in terms of a physical distance that you traverse. And so I think this is an additional piece of information which is actually not accessible in the conventional brain graphs that I showed you before, but that we can now start to exploit as well. Obviously, major disadvantage is that these adjacency matrices become huge, but from the other hand, they're really sparse because the only connectivity they have is very local connectivity. And so that makes them still quite manageable. If you're interested in computing their eigenvectors, you can still do that too. Not all of them, of course, but you can still get leading eigenvectors very easily with a large-scale eigensolver. And I can show you just a few examples, because now visualizing these eigenmodes is really directly mapping onto the brain, because every node is a voxel, so I can actually simply plot brain volumes for you. So this is the first eigenmode, which is a kind of a constant, but already very nicely revealing the dominant structure within the white matter. And then you can see that now we have the first non-zero eigenmode, which is now anterior-posterior pattern. Then we have the left-right hemisphere. And then we start to have, just like before, more complex combinations and details that show up from this very high-resolution connector. And I think there are quite a lot of applications we can pursue with this high-resolution brain graph. One of them we have been tinkering around is to formulate a graph signal recovery problem where we consider the graph signal that we get from fMRI data only on the nodes that are located in the gray matter, because that's the only location where basically both signal would make sense and is certainly strong enough to be useful. And so that means that we don't have any signal on the nodes that lie in the white matter. And so now the idea would be, through graph signal processing, to formulate basically what is called sometimes an in-painting problem, where we want to get a signal that is on the gray matter, fitting with the observations, with the bold signal, but in the white matter, respecting the smoothness on the graph, and therefore kind of reproducing a plausible activity pattern on the white matter, given the measurements on the gray matter. And so this reverts to an iterative implementation, which is a kind of a diffusion under constraints, which we can then regularize and so forth. And so this turns our fMRI measurements into measurements that now have information on the white matter, and that we can use subsequently to actually look how white matter structure is mediating gray matter activity that was measured in the original data. So I think in terms of neuroscience, they're very interesting questions we could subsequently answer with this. And finally, I think there are still a lot of opportunities also by the ongoing and future advances in magnetic resonance imaging. In particular, for instance, there is this layer fMRI, which is becoming increasingly feasible with ultra-high field 70 scanners, where we start to be able to measure layer-specific functional signals, which means that within the gray matter, we start to discriminate between signals that are originating from different layers. And depending on the region in the brain, we know that some layers are more processing the input and others are producing the output. So this means that if we combine this knowledge, we could ultimately get to probably directional brain graphs, which of course would not only be more of a challenge in terms of GSP, but then also in terms of insights from the neuroscience be extremely interesting. And there is more than the brain, there is more than the brain, because it's also possible to do MRI and fMRI of the spinal cord, not only the cervical part, but also the thoracic and lumbar. And so we have now since a while started to look in that in the lab. And in particular, we have a new project where we are trying to look into combined MRI and fMRI of the brain and the cervical spine to basically investigate how these two parts of the central nervous system are interacting during task, during rest, and actually also come up with the first brain-spine type of connectomes, which I think is interesting again, both for neuroscience and for graph signal processing. And so that brings me at the end of the talk, and I certainly need to thank the whole lab, in particular the people on this slide who contributed a lot to all the graph signal processing work that I could show you. We also have some implementations on GitHub, and we are working on a library for GSP for neuroimaging, which you're happy you're happy to try out or to reach out to me if you have any questions for support. So thank you very much. I'm always happy to answer questions if there are any. Thank you very much.
Yeah, so thanks, Dimitri, for the very interesting talk. So I think that we should open the floor for questions now. But before that, I think there was one in the chats already that was posted during the talk. So question from Perugu. So he was wondering, like, how do we choose the lambda to decide about the high pass and low pass regions?
So I think he's probably referring to the cutoff frequency. So what we do is we compute the graph energy spectral density over all the subjects we have, and then we compute the cutoff as where on average we have 50% of the low pass energy. So it's the median energy you want across subjects. So on average, the low pass is capturing the first 50% of the energy and the high pass the next 50%.
I see. So it varies from the set of subjects from another set, right? So it depends on the set of subjects you encounter.
Yes, but this is about 100 subjects, so it's actually quite a lot already. I mean, we have a very good effort.
I mean, just out of my own curiosity, so is there like any differences you see when the demographies of the subjects are changing?
We didn't look into that. It's an interesting question. I mean, I can tell you, I mean, given the results we have on the brain fingerprinting, so the fact that this SDI map is actually specific somehow for an individual as well, I would say yes. I mean, I think there should be a lot of information in there, and not just, let's say, things that are unique for the individual, but also things that relate to personality traits to a certain extent, or neuropsychological scores, and also disease, or early disease, late disease. So and there is actually Enrico Amico in the team, who's our fingerprinting expert, and he has already some results where he's showing this idea, but not using SDI, but another type of measure, but still to do fingerprinting. And he's showing what's the influence of mild cognitive impairment as the first onset to Alzheimer's disease, for instance.
Yeah, that's very interesting. So, well, I guess we should open the floor for others for questions. So, I mean, in case you have questions for Dimitri, feel free to just unmute and ask your questions.
Maybe I have a question. Thanks, Dimitri, for this talk. So I'm just, I have a question regarding the very last part of your talk, that is a combination with the spinal, the project that you have in combining both parts of the body. So do you consider there heterogeneous graphs, or what is the definition of the node in that case?
So you are trying to combine the spinal cord with the null model topic, or
no, the spinal cord. So there you mentioned that you are trying to combine the brain.
Yes.
With the spinal part. So what is it? What is the definition? I was wondering what's the definition of the graph there, because in the brain I can understand what is the definition of the graph. But there, are we talking about two different graphs, or it's an?
Yes, I mean, it's a very good question, and so you're a bit suffering with it now. So, I mean, there is, in terms of a structural connectome, there is not much established at the level of the spinal cord. So we do have knowledge from anatomy, let's say from animals and dissection in humans, autopsies basically, but yeah, there's still many open questions, and the data you can have from diffusion weighted MRI is not as great as for the brain to give you, these very nice streamlines where you can do more easily tractography, and so let's say get the equivalent of a structural connectome. So at the moment we are probing different options how we would continue with that. What, from the other hand, is certainly feasible is to get high quality functional data out of the spinal cord. And so there we have made, I think, good progress. So we are able to characterize ongoing activity both at rest and at task in the spinal cord in terms of meaningful contributions of different anatomical parts within each level and between the levels of the spinal cord. And so one of the striking things is that for sure, the spinal cord is not just a relay station between the brain and the muscles, the periphery. It is doing much more. It is probably much more heavily involved in proprioception, in continuous monitoring of your limbs, of your sensations, and that is requiring a lot of neural activity at the level of the spinal cord. So the resting state spinal cord activity is certainly, I would say, as rich as we have discovered 10 years ago in the brain. And I would expect a bit of the same, the same line of research coming in over the next years at the level of spinal cord. It is much more interesting than often supposed. Let's put it like that. But the connectome itself is still a bit of a challenge. Absolutely agree on that.
Thanks. Yeah, happy to discuss it offline as well. Thanks.
Yeah, so I think before we before we ask the questions on the floor, so I guess there's a few questions in the chat that just appears. So Manuela, are you here? So I think it's okay if you can also just unmute and ask.
Yes, I'm here.
Yes.
So thank you very much for your talk, Dimitri. I was wondering whether do you think that the structural decoupling index could be also used as a biomarker for disentangled pathological traits, such as in brain tumors patients or stroke, where there are huge lesions?
Yes, I think so. So as I mentioned, Enrico, he has been working on fingerprinting using functional connectomes. So not yet SDI, but functional connectomes. And he has shown that those fingerprints actually can be meaningfully related to mild cognitive impairment and Alzheimer. We have an ongoing project where we want to look at stroke specifically, but we don't have results yet, so it's a bit too early. So I think, yeah, they are kind of a biomarker, or if you, that could play, that could somehow summarize in a meaningful way brain activity, and related also with the underlying anatomy and connectome. We actually have one paper on a cohort of 22q11 subjects. So this is a genetic deletion syndrome, and those subjects, they have a very high risk of developing schizophrenia. And there we showed that the SDI map is also very different between this cohort of patients and, let's say, control group, in regions that are actually known to be, let's say, different, altered in this population. So there is certainly interactions between the activity and the anatomy that are meaningfully also changed and reflect into different SDI values.
Okay, thank you very much.
Yeah, so I think the next question is from Wayman. So are you around, Wayman? Okay, so if he's not around, then maybe I can just ask his questions here. So he's asking, like, can you discriminate synergistic from antagonistic brain activities?
Okay, so I think, Raymond, feel free to unmute if you're there. I guess you refer to activity being more synchronized versus being more segregated in a way. So, I mean, the answer is yes, but we did not do this yet with GSP. So we have another line of research where we look really into the dynamics of fMRI signals without the GSP framework. And there we exactly, let's say, see these differences when brain activity is more synergistic versus antagonistic, and all these dynamics that develop over time. So one of the open topics is to combine this now with GSP, because in the end, what I showed you so far in terms of temporal characteristics, we just computed energy over time, right? So we did something very simple for now, and we can bring in much more specialized and advanced measures to characterize the temporal domain. And this is certainly something we want to do. And this is also a line of research to continue, for sure.
Yeah, so I hope that this addresses Wayman's question. So well, let's open the floor again. So if there's any other questions for Dimitri.
Yeah, maybe I have a question. So thanks for the nice talk, Dimitri. So I was wondering, so you look at low pass versus high pass. So could you consider also more bands and more frequency bands, and would that be useful somehow?
Yeah, very good question. I think, but this is a bit of a gut feeling for now. So we tried different variations so far. So first of all, we tried to use, instead of the ideal low pass, something that is a little bit more, let's say, elegant, like a diffusion filter, for instance, for the low pass, and then the complement of the diffusion filter for the high pass, because I think, it has a more easy interpretation somehow. And this gives very similar results. So the SDI maps basically look more or less identical, which is good news, right? And yeah, it's then more interpretable. Now, about having something that is more maybe subband type, you could go towards wavelet type of transforms even. My current take on it, and that's where my gut feeling comes a bit in from the results I've seen. I think with the connectomes on the atlas with about 360 regions, like what I showed you now, I think we don't have enough regions to do that. But I think with the high resolution brain graphs, we actually will have meaningful partitioning in the spectral domain that goes beyond simple low pass and high pass. And actually, you know what, I mean, this is one of our really ongoing challenges, which I hope we will soon be able to crank out, is to somehow have the equivalent of a scale that relates to spatial distance. Because then we can, and for this we will need the high resolution brain graph. We will need exactly this property that I mentioned, that you somehow have a relationship between the number of hubs on the graph and the physical distance. And I think we're almost there, and then we would be able to say to what extent is a certain activity pattern relates to a certain spatial scale. And that would bring us closer to your question.
Okay, yeah, thanks. Interesting.